So our next topic is going to be organic compounds. And if y'all remember from your freshman year, we had 4 major classes of organic compounds. Remember, proteins, carbohydrates, lipids, and nucleic acids. If y'all remember, our strict definition of an organic compound is that it has a carbon-hydrogen bond. Okay, so it doesn't just have to have carbon. Okay, it has to have a carbon-hydrogen bond. Like, for example, carbon dioxide is not an organic compound because it does not have a carbon-hydrogen bond. Okay, we're going to spend just a couple minutes here at the beginning talking about what are called functional groups. You do not have to memorize all of these functional groups. What I need, and for example, you do not need to memorize that a carbonyl group here is a C double bonded to an O. You do need to be familiar with their properties and how they impact the molecule. Because the reason why we're going to talk about these functional groups is these functional groups will have an impact on the behavior of the molecule. They may make it polar, they may make it easier for it to dissociate, they may affect its structure. Okay? So we're just going to kind of run through these functional groups very quickly. Our first functional group we're going to look at here is the hydroxyl group. Okay, so a hydroxyl group is going to be also called an alcohol. Okay, and so their suffix there will usually be OL. Like, for example, here you have ethanol, that will end in the OL. Okay, these hydroxyl groups, okay, so you're going to have an OH that is added on to your carbon chain. And due to that OH ion, it is going to make them polar. Okay, and so remember, oxygen is significantly more electronegative. Remember, that's what makes water polar. And so by having this oxygen molecule here, that's going to make this whole thing it's going to make that whole thing polar. And so because it makes the whole molecule polar, okay, it is able to form hydrogen bonds with the water. And when molecules are able to form hydrogen bonds with water, they can help dissolve things. For example, alcohols can dissolve other organic compounds like sugars. They could help dissolve proteins. Okay, because remember that polarity allows them to bond with other molecules. Okay, so that's an important thing that you'll need to remember about alcohols. Okay, is that they offer this polarity. Next, we have what is called a carbonyl group. Okay, so our carbonyl group is a carbon double bonded to an oxygen. Okay, so it is a carbon double bonded to an oxygen. We have 2 basic kinds. Okay, we have a ketone versus an aldehyde. Aldehydes would be, for example, this one down here would be an aldehyde when the carbon-oxygen double bond is at the end of the molecule. This one here would be an example of a ketone where that carbon-oxygen bond is there in the middle of the molecule. Okay, right here, it talks about ketones and aldehydes being structural isomers. We haven't really talked about isomers. All you really need to know for that is that a structural isomer, it's just going to have a different structure. It has the same formula. Like if you look here, we've got 3 carbons in both of these. Okay, we've got 6 hydrogens in both of these. Okay, and we just have the 1 oxygen in both of them. And so my formula is the same, but my arrangement is different. So they're structurally different, structural isomers. Okay, so one of the things that you would need to keep in mind, again, with our carbonyl groups, okay, is that these are found very commonly in sugars. Okay, and again, having that oxygen there can alter the polarity of the molecule. Next, we have a carboxyl group. Okay, so a carboxyl group is going to be a little bit different than a carbonyl group because it's going to have this OH bonded to it as well. Now, your carbonyl group was just your C double bonded to the O. Your carboxyl group is going to be a C double bonded to an oxygen as well as to that hydroxyl group. A lot of times you'll see it written like this. Okay, that is a carboxyl group, sometimes called an organic acid. So this is going to act as an acid because this hydrogen here can dissociate. And so when that hydrogen dissociates here, when it's ionized here, okay, that's going to obviously increase our hydrogen concentration in our solution. So that would make our solution become more acidic. Okay, so there is, it does have properties that will affect the pH, can affect the pH of the solution. Next, I've got an amino group. So an amino group is always going to have, sorry about that. An amino group is always going to have a nitrogen associated with it. Okay, amino groups can act as a base. Okay, and to be able to act as a base, remember they would be able to pick up a hydrogen ion. Okay, because remember bases help decrease the hydrogen ion concentration in the solution. So these can act as a base, and so you can see here when it has been ionized, because see it has a positive charge because it has that extra hydrogen, it is acting as a base then. Okay, if y'all remember right here, glycine, so this would be an example of an amino acid. Remember all of our amino acids, they have this amine group. Okay, they have this one, do you recognize it? And that would be a carboxyl group. And then remember they'll have that R or that variable group. Now on to our sulfhydryl groups. Okay, so these are going to contain sulfur. You can see right here we've got our sulfur with a hydrogen here. Okay, they are also called thiols. Okay, one of the times, really kind of the only time we'll really focus on this, is how this affects a protein's structure. Okay, these sulfhydryl groups will, they have an important role in determining the structure of a protein. Their interactions with other functional groups help determine that shape of a protein, the conformation of the protein. And if y'all remember, the shape of a protein is everything. Okay. Our next to last group here is a phosphate group. Okay, phosphate groups are sometimes also called organic phosphates. They're obviously going to have a phosphate associated with them. You guys would have seen these before your freshman year in phospholipids. They contain a phosphate group, and we'll talk more about ATP and its phosphate groups. Okay, so phosphate groups are going to contribute a negative charge to the molecule. Okay, and so that can have obviously is going to have an impact on the molecule. Okay, phosphate groups are definitely going to be able to interact with water because water is polar, and we've got a charge going on here. Okay, and one of the ways that they can interact with water is they can release energy. And again, we'll talk about ATP and how that works later. So our last functional group here is a methyl group. Okay, so with our methyl groups, okay, methane is CH4. Okay, there'd be an extra hydrogen there to fill all of your carbon bonds. Okay, so our methyl group is just a carbon-hydrogen compound attached on the end. Okay, methylation plays a huge role in DNA and gene expression. That's one of the things we will talk about. One of the ways we can help regulate genes are expressed is by methylating the DNA. It can turn genes on and turn genes off. Okay, so methyl groups, even though they seem very simple, can have a huge impact on an organism. Okay, so remember, you don't need to memorize all of the conformations of these. You don't need to remember that methyl groups are CH3 or carboxyl groups are COOH, but you do need to have an idea of their properties and how they can impact the your compounds, your organic compounds. Because the rest of our video, we're going to be hopefully mostly reviewing our organic compounds, our proteins, lipids, nucleic acids, and which one do they have? Carbohydrates. Okay, so the rest of our video here is going to be about organic compounds. If y'all remember from your freshman year, there are 4 basic types. We've got our carbohydrates, our lipids, our proteins, and our nucleic acids. Most of this should be review from your freshman year, so we're going to try to go through it relatively quickly. Okay, the first thing we talk about is how these things are formed. Okay, y'all remember the difference between a monomer versus a polymer. Okay, remember monomers are the little ones that make up the larger polymer. Like if you had a set of Legos and you were building a castle, your castle would be your polymer, each individual Lego would be your monomer. And what we're going to talk about is how those are joined together first. Okay, and so they're going to be joined together as a general rule using what are called dehydration reactions. So with a dehydration reaction, we're going to be removing some water, okay, just like it sounds. So we dehydrate it, we remove some water, and that will actually allow the monomers to link together. Hydrolysis reactions, on the other hand, okay, they are a lysis reaction. So remember, lysis means to break. Okay, and we are using water. Okay, so the hydro part is the water. So when I add water, okay, it will break the bonds. So it'll take the polymer and break it into the smaller monomers. So let's look at a couple examples of these. So our first example here is a dehydration reaction, or what is sometimes called a dehydration synthesis, because it's making something new here. I'm synthesizing something. So with my dehydration reaction here, okay, I've got my unlinked monomer, I've got the beginnings of my polymer, so there's my short polymer, okay, and so I dehydrate or remove a water molecule, which allows these two to bond, right? If I remove this hydrogen, remove this hydroxyl group, this hydrogen would be positive on its own, the OH group would be negative, so those two would attract to each other to form the water, and this short polymer then would have a slightly negative charge on it, and this unlinked monomer here would be positive. So those two would be attracted to one another, which would allow it to form that bond, allow something to be added and elongate the monomer. So our dehydration synthesis removes water to be able to link, to be able to make bigger molecules. So our hydrolysis reaction will be the opposite of our dehydration reaction. So here in my hydrolysis reaction, I've got my long polymer here, and when I add water to the polymer, that water is going to break the bond. And so that water will split, the hydrogen will join here, the OH group will join there, but I'm splitting the polymer, so I've broken this polymer into two different parts, by adding a water molecule. So with my hydrolysis, I break down the polymers by adding water. Our first class of organic compounds would be carbohydrates. Okay, and if y'all remember, carbohydrates are also our sugars. Okay, so carbohydrates, if you see sugar, also the suffix "ose," like glucose, sucrose, fructose, cellulose. Okay, so sugars or that suffix "ose" should make you think carbohydrates. Okay, our monomer of our carbohydrates, those are monosaccharides. The polymer here would be a polysaccharide. The example we have down here, okay, this will be a monomer. This here is glucose. Okay, and so you can see it's a ring shape. Okay, it's a 6-carbon ring, 12 hydrogens, okay, and it's got 6 oxygens. So some examples of my monomers of carbohydrates: I've got glucose, fructose, galactose. Okay, our monomers, our carbohydrates, are usually found in our 1-to-1 ratio. Okay, that's 1 carbon to every 2 hydrogens and 1 oxygen. Okay, and so that ratio is usually pretty consistent. You can see that happening here with our 1, 2, 3, 4, 5, 6 carbons. Okay, you'll count there'll be 12 hydrogens and 6 oxygens. And like I mentioned before, our monomers here, our monosaccharides, are also usually ring-shaped. Okay, so let's take a look at what a disaccharide would look like. And this here is a disaccharide. Saccharide, also, you need to be thinking sugar or carbohydrate. Di is 2. So with this disaccharide here, I have 2 monomers, okay, and they are linked together, okay, by what's called a glycosidic link. Okay, so basically I have 2 glucose molecules here, and they are held together by this glycosidic linkage. If you remember from our previous slide, these glucose molecules look a little bit different. Okay, they don't have all the carbons laid out, but everywhere there's a corner, okay, there is a carbon there. So each one of these corners would represent a carbon. And so when I look at the formula here for sucrose, so this disaccharide here is sucrose, which is the sugar you eat. Okay, it's the white sugar, that we bake with, we eat. Okay, so the sucrose formula here is C12H22O11. So if you notice, that's a little bit off from our 1-to-1 ratio that we said our monomers have, but that's because we had to form this bond here. And if we think back to how most of our polymers are formed, they're formed by that dehydration synthesis reaction. So I had to lose a water here. And so if I lost a water, okay, I've lost 2 hydrogens, so that's where my 24 got down to a 22, and I lost 1 oxygen. So that's when my oxygen is down from a 12 to a 1. All right, let's look at our first polysaccharide. So a polysaccharide is going to be many sugars. Okay, so many monomers, many monosaccharides put together. So our first polysaccharide we're going to look at is starch. Okay, and so starch is how energy is stored in plants, how extra glucose is stored in plants. I have been having trouble getting my starch picture to upload, but all starch is a long chain of glucose molecules. And so what I have here is I have multiple glucose molecules here. And with starch, these glucose molecules, we lose that hydroxyl group here, we lose this hydrogen, and so they form a link there. They form that glycosidic linkage. I've got my dehydration synthesis. Okay, so I'm losing this hydroxyl group, I'm losing that hydrogen, and they'll link together in a nice straight line like that. And that's where we're going to store the extra sugar. Okay, it's in the form of starch. Our next polysaccharide is glycogen. Glycogen is going to be found in animals. So glycogen is how animals will store their extra glucose. You'll see here's the linkages here. So it looks a little bit different. Okay, it's not such a straight line like starch was in the plants. Okay, we find this mainly in the liver and muscle cells of animals. Okay, and when our glycogen storages are full, we'll then store that extra sugar as fat. And you can see the glycogen granules here in this microscope picture surrounding the mitochondria. The next polysaccharide that we're looking at here is structural. If y'all remember back from your freshman year, some of the jobs of carbohydrates were to, some of them were structural, and a lot of them were for short-term energy, very quick, rapidly available energy, broken down very fast. Okay, so the next two polysaccharides we'll look at are structural. So we've got cellulose here, and then we'll look at chitin. So remember, cellulose, there's your ose for your sugar. Okay, cellulose is what's in the cell wall of plants, so it is definitely structural. Okay, lots of organisms, eat plants, but they cannot break them down because they don't have the enzyme to do it. An enzyme called cellulase to break them down. Okay, and that's where some of your mutualistic relationships will come into play with, for example, the microbes that are in your intestines. They help to digest the cellulose, some of the cellulose that you have. Termites, a lot of herbivores, they're going to have these mutualistic relationships where these microbes get a place to live and they receive food, and the organism the host organism is getting the benefit of the breakdown of the cellulose. In humans, our diet in cellulose is considered our insoluble fiber. But you can see structurally, it's a little bit different. The linkages here are a little bit different than they were in starch, which is also found in plants. I still have glucose monomers. I still have these monosaccharide monomers. But you'll notice this alternating linkage is here, which allows it to provide the structural support that starch would not. Our last polysaccharide that we're going to talk about here is chitin. Chitin is not, remember, it's not chitin. Chitin is another structural polysaccharide. You're going to find it in the exoskeletons of bugs, of arthropods. And you would also find it in the cell walls of fungus. Mushrooms, they have this, their cell wall contains this structural polysaccharide chitin. Okay, over here is just an example of humans using it for medical purposes. Okay, but very similar to cellulose in the fact that the arrangement of these monomers here are going to provide structural support. So moving on to our next group of organic compounds, we've got lipids. Lipids are hydrophobic, and if y'all remember, hydrophobic would be that they are water, quote-unquote, fearing. Okay, so these are nonpolar. That's what makes them hydrophobic. Wow, my pen is really large. Okay, so they are nonpolar, so they don't have any kind of charge in them. And they don't because they are made up mostly of, Okay, they don't because they are made up mostly of, what is that? There we go. They are made up mostly of carbon-hydrogen chains, okay, and so they do not form monomers. Okay, they don't one, or they don't form polymers. They don't have one particular monomer. We've got a really wide variety of molecules here with lipids. We're going to focus on 3 of them. Okay, we are going to focus on, if I can bring it, they're not coming up. Okay, we're going to focus on fats, phospholipids, and steroids. And we're going to talk about each one of them individually. But these all have very different structures, okay, in the sense that, your phospholipids are going to have a phosphate group, your fats are going to be made of glycerols and fatty acids, your steroids are going to have ring-shaped structures. So there's not one particular monomer for these lipids. And like I said, they are all hydrophobic because they are made mostly of carbon-hydrogen bonds, which will make them non-polar. So our first one we're going to talk about is fats. Fats are also called triglycerides. Okay, so let's get that spelled for you up there. Okay, so fats are called triglycerides because they're going to be made of one glycerol molecule and 3 fatty acid molecules. So over here I've got my glycerol molecule right here, which is made of a carbon skeleton with 3 alcohol groups attached to this carbon skeleton. Remember alcohol groups from our functional groups before? Okay, and there's going to be 3 fatty acids that will attach to it. So there's our big long carbon hydrogen skeleton there in the fatty acid. So these fatty acids will attach to the glycerol molecule doing this dehydration synthesis. So we remove the water molecule and we will end up attaching 3 fatty acids to get a completed fat or triglyceride. So I've got my glycerol molecule here, 3 ester linkages, which were formed by dehydration synthesis. So if I wanted to break this fat down, okay, I would do a hydrolysis reaction, right? And I would need to add water. I would need to add 3 molecules of water to be able to break that down. Okay, this particular fat is what is considered saturated. We're going to talk about that on the next slide. You'll see some more pictures of it. But I want to be able to show you this here. It is saturated. These fatty acids are saturated because you'll notice there are no double bonds in them. They are all single bonds. So every single place that a hydrogen could bond, there is a hydrogen bonding to it, bonding to these carbons. And that's what makes them saturated. They're fully saturated with hydrogens. So your saturated fats are mostly animal fats, okay, or animal products. The example you hear is butter. Milk would be an example of a saturated fat. These are completely saturated with hydrogen, like we said before. So their fatty acids are in a straight chain, which makes them solid at room temperature. Your unsaturated fats are usually plant-based products, okay, like your olive oils. Okay, so these are usually plant-based products. They're going to have at least one double bond somewhere in one of those fatty acids. And that double bond causes this bend or the kink in the molecule. And when they're bent like this, they cannot, the molecules can't get as close together, and so they are usually liquid at room temperature. On a little nutritional segue here, y'all probably hear a lot about trans fats, especially because a lot of restaurants and food labels, they're being asked to show their trans fats, and you'll see commercials about we have less trans fat than the other guy. And so let's talk just for a second about what trans fat is. Okay, so trans fats are fats where these hydrogens, okay, are on the opposite sides here. That's what trans means, is across. And this would be a cis configuration there, where they're on the same side. Okay, so our bodies don't have enzymes really to break down trans fats very well. And so because we have a lot more of the enzymes that can break down these cis fats, so our body is much better at breaking down cis orientation fats. And what happens when, with a lot of processed food, okay, is we go through this process, the food goes through this process that's called hydrogenation, where we're adding hydrogens to help saturate the fat, to help make it stay solid at room temperature. For example, peanut butter. If you've ever had like natural-based peanut butter, usually there's oil on top that you have to mix in because those oils have not been hydrogenated, so they don't stay solid at room temperature. Okay, and as a general rule, the reason why most of our food is hydrogenated is because of appearance. People don't like to see oil on the top of their peanut butter. They don't want to have to stir it in. But these hydrogenated fats, a lot of times when we hydrogenate things, they end up in this trans configuration. Okay? And it also adds that hydrogenation of adding more hydrogen makes it harder for us to digest these things. Okay? And so too much of these can build up in our system, which can cause disease and other problems. We also have what are called essential fats. Essential fats are not synthesized by the body, but they are required. There's things like your omega-3s. Okay? These are required for normal growth and function. You need some what is called quote-unquote good fat. Okay, so still talking about fat, so this is a picture of what's called adipose tissue. Okay, so adipose tissue is your fancy name for your fat cells or your fat tissue. So you can see in these cells, they're all full of fat. Okay, and so what this does, the adipose tissue, is it gives us our long-term energy storage. It helps with insulation. You know, fat has a high water content, so it helps with insulation. It helps maintain body temperature. It also helps with cushion and protection. If you think about your body, for example, your kidneys. Your kidneys don't really have a lot of bony protection. They sit in essentially a giant bed of fat. And so that fat is what helps keep them in place when they're embedded in your back. They help keep them in place, and they help support them. That's why when you see boxers, when they're punching for their lower back there, they're going for the kidneys. So our next lipid example is a phospholipid, okay? And it gets its name because obviously it has a phosphate group right here, okay? And then it has a fatty acid tail. And if you'll notice in this picture over here of the bilayer, your tail is hydrophobic, where your head is hydrophilic. Okay, so if you remember phosphate groups, they will have a charge on them, and so they are hydrophilic, they are water-loving. Okay, but the tail is that big, like we saw on the triglyceride, that fatty acid tail is a big long chain of carbons and hydrogens, and so those do not really have a charge, they are nonpolar. So that'll make this molecule what's called amphipathic. Okay, and amphipathic means that they have both hydrophilic and hydrophobic parts on them. So when these phospholipids end up in a water medium, which is what most biology is in, right? Our cells are water-based, the fluid that surrounds the cells are water-based. So when these amphipathic molecules end up in a water-based solution, they will orient themselves in a way so that the hydrophobic tails are there and towards the middle, quote-unquote protected from the water, and the hydrophilic heads are on the outside where they have that attraction to the water. And this helps form a natural barrier for us. Remember, phospholipids are what make up the majority of your cell membrane. So our last lipid example is a steroid. Okay, and so all of your steroids have the same basic structure. Okay, they're going to have this 4-ring structure right here, and then this carbon skeleton is what will be different from them. Okay, what we normally think when we think of steroids would be like anabolic steroids, when you athletes that take these steroids. Okay, because what that is synthetic testosterone. Because steroids will help provide the basis for some of our hormone structure. We have lipid-based hormones and we have protein-based hormones. And steroid hormones, 2 examples would be testosterone and estrogen. And they are basically exactly the same. They have this same 4-ring basic structure, but this carbon skeleton up top, that is what's going to be different. That's what's going to make male hormones versus female hormones. Steroids are also important to our cell membrane. Okay, they help form the basis of cholesterol. And usually we hear about cholesterol being bad and it makes us have heart disease and it's all awful and horrible. But that is not true. Not all cholesterol is bad. We do have cholesterol helps in our cell membrane. It helps the cell membrane stay fluid and flexible. Okay, so basically what we're trying to get across at this point now is that all of our lipids are not bad. Okay, all lipids or fats, they are not bad. Okay, so let's shift gears then to our next organic compound. Our next organic compound would be our proteins. Monomers of our proteins are amino acids. They have a wide variety of functions. We're going to go through a number of the functions. Also remember that our enzymes are proteins. Okay? Enzymes speed up chemical reactions. Your next video will be over enzymes, and we're going to finally get to do a big lab, like an in-class real lab, over enzymes coming up later this week. Or I guess this next week. So our proteins... Okay, one of the functions here is for structure or support. Whoops. Okay, the examples they give here are keratin. Keratin is the protein that you find in your skin. Okay, it's what makes your skin kind of water resistant, waterproof. It's the protein that's in your hair. It's in horns, feathers. Okay, it's in fingernails. You also have structural or support proteins that you find in silk. that spiders and insects use to make webs and cocoons. Also, connective tissue, in particular collagen and elastin, has a lot of structural proteins in it. You'll see a lot of collagen and elastin in your tendons and your ligaments, in the tissue underneath your skin that help support your body. They help hold your bones together. They help hold your muscles to your bones. When this connective tissue, as we get older, and this collagen in particular starts to break down, that's why we get wrinkly. Okay? So protein can definitely offer structural support. Proteins can also help act as storage conduits, especially for amino acids. Remember, our monomer for proteins is amino acids, so they're going to help store amino acids. For example, casein, the protein that's found in milk. Okay, baby mammals start off drinking milk only, and so they need a source of protein. They need a source of amino acids to be able to build their own proteins. And so the casein in the milk is the source of those amino acids to be able to build their own proteins. Plants store proteins in their seeds. Another example here is egg white. Okay, egg white is, we talk about eggs being very high in protein because the egg white is the amino acid source for the developing bird embryo that's inside of this egg. Okay, proteins can also act as transports for substances. The example you see here, you've got all these molecules on the outside of the cell, and they're going through that protein channel to get to the inside of the cell. If y'all remember from your freshman year, we talked about facilitated diffusion. Okay, remember that was the people that needed help. If y'all were in my class and we talked about the river analogy, these people that need a raft to be able to float down river because they sink to the bottom. Doesn't require any energy. They still go with the concentration gradient, but the molecule is either too charged or too big to go through that phospholipid bilayer. And so we have these larger proteins that can act as transport. The other example they list here is hemoglobin. Okay, hemoglobin is the protein that we find in our blood that helps transport the oxygen to our various tissues, various cells. Okay, we also have what are called receptor proteins. So receptor proteins are going to help respond to chemical stimuli. For example, let's say this was a neuron. Let's say it's a neuromuscular junction. So that's a neuron, and this is my muscle. Okay, my neuron is releasing my neurotransmitter, all these little red dots, and they're traveling across to bind to these receptor proteins. And you notice the receptor protein is the right size and shape to receive that neurotransmitter. So when that neurotransmitter binds to that receptor protein, that signal then tells your muscle it's time to contract. Okay, so they help with reception and cell communication. Here we've got some proteins that are acting as defense mechanism. Okay, we've got some antibodies here. So all of these little purple substances here, these are our antibodies. They're trying to fight off, in this case, a virus. Over here, there's a larger bacterial cell that they're trying to fight against. Some of the other functions that our proteins have is they help with movement. Our muscles are made of protein, large amounts of protein found in our muscles, so that's going to help us move, as well as hormones. Remember we talked about steroids just a minute ago. I mentioned to you that there are two basic kinds of hormones. There are steroid hormones and protein-based hormones. Okay, so next let's talk about the monomer of proteins a little bit. So our monomers are amino acid. Again, this is most of this video should be reviewed from your freshman year. Okay, so I've got my amino acid over here. Remember, this is the same in every single amino acid. Every single amino acid has an amino group. They have a carboxyl group. Remember our functional groups? Amino groups have a nitrogen. The carboxyl groups, remember those are our COOH. They can make things act like an acid. Remember, it's an amino acid. Okay, and then this is what changes, right? Remember we have that R group or the variable group. That's the one that makes every single amino acid different. Okay, you can have hydrophobic R groups, you can have hydrophilic ones, you can have ones that are electrically charged. These R groups are going to have a big impact on the protein structure, which has a huge impact on the protein's function. So proteins are made by linking these amino acids together. And when we link these amino acids together, we form what's called a peptide bond. Surprise, surprise, we do this again by a dehydration reaction. So in my amino acid, my amine group is called my N-terminus, where my C group, my carboxyl end, is called my C-terminus. And so on two amino acids, these two will be able to join together. I'll be able to lose the OH group off of the C terminus and lose one of the hydrogens off of the N terminus. So there's my dehydration. I've released water, okay? And when I release that water, I form this peptide bond, which is why a protein sometimes is referred to as a polypeptide. A chain of amino acids would be a polypeptide because there would be lots and lots of these peptide bonds that are found between the amino acids. So our protein structure has a huge, huge impact on function. Protein structure and function go hand in hand. Okay? And so our primary structure, there are 4 basic levels to protein structure. And our primary structure is going to be the sequence of the amino acids. What order are these amino acids in? Remember when we did protein synthesis your freshman year? That's the only thing we focused on. Like you can see here, I've got glycine, arginine, valine, alanine. Okay, and so that's all my primary protein structure is what order are these amino acids going to be in? And that's going to be based off of our DNA, right? Our DNA is going to tell us the primary structure. It's going to tell us what amino acids we're going to be making and linking together to make this protein. So after we've established this polypeptide chain, after I've made this long string of amino acids, and I've made this polypeptide chain, so the protein's going to move on to its secondary structure. And the secondary structure is going to be due to hydrogen bonds, and it's going to be hydrogen bonds between the backbone structures. So right through the middle, those carbons and nitrogens that made up my amino acids, that made up my amine group and my carboxyl group and my central carbon right there in the middle, that's my backbone, not my R group. I don't care what my R group is right now. Okay, I'm just focused on the backbone. And so these hydrogen bonds here that are forming between these backbone structures, they're going to start kind of twisting that protein a little bit. And they can twist it either into a helix, which would be called our alpha helix, or into what's called the beta pleated sheet, which kind of looks like if when you make a fan out of paper and you keep folding it back and forth to make that fan, that's what your beta pleated sheet will look like. So after our secondary structure, we're going to move on to our tertiary structure. So with the tertiary structure, this is going to be dependent upon those R groups. So our R groups in the tertiary structure, like I said, they can have a variety of properties. You can have hydrophobic ones, hydrophilic ones, whatever. These are going to help. These interactions are going to really kind of finish up the conformation or shape of the protein. Sometimes you will see that instead of shape or structure, you will see that they'll talk about the conformation of the protein. Okay, they're just really referring to its shape or structure. And so there's lots of different ways that these R groups could interact. They could form ionic bonds, the transfer of the electrons. They could form disulfide bridges, which these are a form of covalent bond, so these are nice and strong. Okay, if you remember back to our functional groups, we had that functional group, the sulfur groups with the thiols. So there's a covalent bond there. They could do the London dispersion forces and IMFs, okay, and have those weak interactions, or the hydrogen bonding between the various R groups, okay, based on those, those things based on the partial charges and the induced dipoles. So these R groups can interact in a variety of ways, but these are really going to kind of finish up the conformation of the structure. An example of how you can kind of wrap your brain around this is if you compare curly hair to straight hair. So people that have curly hair have way more of these disulfide bridges. Lots and lots of these disulfide bridges. And you notice it makes this kink, okay, as it forms that bridge, and so their hair looks curly. And while people that have curly hair, if they want it to be straight, what do to it? Well, they start taking heat to it, right? They take a flat iron, they take their brush and their hair dryer, and when they do that, if y'all remember back from your freshman year, heat destroys proteins. And so it ends up, that heat will break those bonds, those disulfide bridges, and so the hair will relax and become, it won't be so kinked here, and it will look straight, and it will be straight. The opposite occurs when someone gets a perm. Okay, they put the rollers on their hair to induce the shape, okay, and then they put that basic or alkaline solution on it, which encourages the formation of these disulfide bridges, and so their hair becomes curly. So the last level of structure on a protein would be the quaternary structure. And all proteins do not necessarily have this structure. These are for bigger, larger proteins that have multiple polypeptide chains involved. The example, the two pictures we have here, I've got collagen, okay, and you see there's two turquoise polypeptide chains and 2 purple ones. So all 4 of those polypeptide chains are interacting together, and that's their quaternary structure, how they're interacting together. If you look down here at the collagen example, I've got 3 different ribbons of 3 different polypeptide chains. And remember, collagen is found a lot in connective tissue, so it's very strong, it's very supportive. And so basically these 3 polypeptide chains, their quaternary structure involves them essentially kind of twisting around one another like a rope, so they get even stronger. So this is just kind of an overview of all of our, just kind of summarizing all of our different structure levels here. So you've got your primary structure, which is just your order, your secondary structure, which is on your backbone interactions, your hydrogen bonds between your backbone molecules, your tertiary structure, which is going to be based on your interactions between your R groups, and then your quaternary structures that are based upon if I'm using more than one of these polypeptide chains. So I've got more than one chain that's gone through these first 3, and those chains start interacting together. So let's look at a couple of examples of what happens or things that could happen to the protein if we do alter its structure, because its function is so dependent upon its structure. So the first thing is denaturation. We talked about enzymes in your freshman year. We'll talk about it again with enzymes in a little bit. Okay, but if you look at your examples here, here's my normal protein gone through its tertiary structure. Its conformation is nice and tight and folded. Here's my denatured protein. So things are, the conformation has changed. It's more relaxed. That protein is inactive. Okay, it will not work because it has lost its shape. Okay, when we heat enzymes, there's a lot of things that can do that to the protein. Temperature can do that. Salt concentration can do that. Acid-base levels, pH levels can denature or make that protein lose its conformation. Another example here would be sickle cell anemia. So if I look at my normal hemoglobin versus my sickle cell hemoglobin, hopefully you notice there's an alteration there in the primary structure. Okay? This number 6 here, instead of being glutamine, is valine. By changing that one amino acid, it exposes this hydrophobic region in the beta subunit. So there is a difference in the folding of the protein, the conformation of the protein, because valine's R group is going to be different than glutamine's R group. So when they fold together there, the shape of the protein ends up essentially being wrong. And so when its shape is wrong, it does not allow it to carry oxygen as efficiently, and it also does not allow it to pass through the blood vessels as efficiently. And so people that have sickle cell will suffer, sickle cell attacks are usually painful. There's a decrease in oxygen supply sometimes to the tissue, and if that's severe enough, the tissue could end up dying. So just by altering that one amino acid out of hundreds of amino acids, you end up with a potentially very serious disease. Okay, our last organic compound are our nucleic acids. Okay, again, hopefully all mostly review from your freshman year. So if you remember, our nucleic acid, our monomer of nucleic acid is a nucleotide. And your nucleotides, whether they're DNA or RNA, remember they're going to be made up of a sugar, a pentose or a 5-carbon sugar. They're going to be made up of a phosphate group, and they're going to be made up of a nitrogen base. And if y'all remember, you had your essentially 5 nitrogen bases. Adenine, thymine, cytosine, guanine for DNA, adenine, uracil, cytosine, guanine for RNA. Okay, our functions of our nucleic acids, they're going to be to store, transmit, and express our genetic info. Remember, examples of nucleic acids would be DNA and RNA. DNA is going to help store that information. RNA is going to help transmit and express it. Remember, DNA has our instructions. RNA is the one that's actually going to carry out and do the work. So let's do a little comparison between the two. Let's see if I can drag these in. So let's talk a little bit about let's first talk a little bit about what these two have in common. Okay, so in common, you're going to find both of them in the nucleus. In particular, with RNA, you're going to find messenger RNA in the nucleus. Remember, DNA cannot leave the nucleus, and messenger RNA is in the nucleus. In protein synthesis, it will eventually leave the nucleus, but does start in the nucleus. You also find the rRNA, the building blocks for the ribosomes. You also find that in the nucleus as well. The monomer for both of these is a nucleotide. Going to have that sugar, phosphate, nitrogen base. So the monomer of both will be a nucleotide, and they're all going to contain adenine, cytosine, and guanine. Now that 4th base will differ between the two. So if y'all remember, DNA will be double-stranded. Okay, so DNA has double-stranded in that double helix shape. Okay, sorry, it had cut something off there. So DNA is double-stranded in that double helix shape. There's only one kind of DNA. it has that nitrogen-based thymine. Okay, it contains our protein synthesis instructions, and it contains the sugar deoxyribose. There's a picture of our sugars down here. Okay. So it's going to contain the sugar deoxyribose. Let's move this up for you. Move that up. Okay, and RNA is going to be single-stranded. Okay, there's 3 types of RNA as compared to the 1 type of DNA. I've got messenger RNA, transfer RNA, and rRNA. RNA is going to be in my cytoplasm and out at the ribosomes in addition to being in the nucleus. It's going to actually do the work and help build the protein. It's going to contain uracil instead of thymine, and its sugar is ribose. Okay, if you look down here at the bottom, you've got your pictures of your 2 sugars. I know they're on the wrong sides, okay, but if you look at them, you've got ribose here, okay, and your deoxyribose here on the other side. Okay, and if you can see your difference, okay, did you guys find it? Okay, here's my difference there, okay, between the 2. Very subtle difference, but makes a very big difference. Okay, so let's take just a minute, look a little bit closer at DNA. Okay, so a couple of things we need to, and this will probably be new to your freshman year, is this concept of DNA being anti-parallel. You may have seen on tests and quizzes that they had this little 3' or 5'. You may have seen something written like this, where you saw the 3', 5', and nobody really explained it to you or talked to you about it. Okay, that's going to be based on our carbons. So if y'all remember your structure of DNA, you know, it's a double strand, and it's made of these sugar phosphate backbone, right? I alternate, okay, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And then on the other side, sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay, and so I have an alternating sugar phosphate backbone with my nitrogen bases in the middle. Right, my genetic information is carried in the order of my nitrogen bases, their sequence that they come in. Well, my 5' end is going to be my phosphate end, where my 3' end is going to be on my sugar end. And that's going to be based on the carbons. If y'all look over here, okay, we number the carbons. Remember I told you the corners are carbons? So there's carbon number 1, 2, and I'm just going around my pentagon here. 1, 2, 3, 4, 5. Okay, so on this one, this is my 5' end because my 5th carbon is out there. And this end would be my 3' end because I have my 3rd, I'm referring there to my 3rd carbon. And we add things only to the 3' end. And that's where I can do my dehydration synthesis. I can remove this OH group, okay, remove a hydrogen from here, okay, and I can attach from a phosphate group on another nucleotide, and I can do my dehydration synthesis. Okay, and so by having that kind of opposite orientation, where you'll notice the 5' end is up here and down here, and then the 3' end is also at opposite sides, okay, that's where we get the anti-parallel. Okay, that's what the anti-parallel means. The last thing I want to do with our DNA is I want to look a little bit at base pairing with DNA. We'll focus with RNA base pairing again with protein synthesis. But hopefully y'all remember your base pairing rules. Remember A to T, C to G. Okay, no exceptions to those rules ever. Okay, so one of the things you may not remember from your freshman year, I know I talked to you about it, but like I said, you may not remember it. Little detail here is our pyrimidines versus our purines. Remember, pyrimidines are 1-ring structures. Purines are 2-ring structures. Okay, let me type this out for you real quick. Okay, here we go. So we've got our little saying up here to remind us of which ones are which. So I've got my pyrimidines are cut from purines. Well, hopefully you will notice that cut is capitalized. And my pyrimidines here are cytosine, uracil, thymine. So pyrimidines are cut from purines. And to be cut from a purine, okay, right, to be cut from something, you started with something large, and you can cut it, right? And my pyrimidines are my 1-ring structure, my purines are my 2-ring structures. Okay, so pyrimidines are cut from purines. Now, pyrimidines are not cut from purines. Okay, just so you know, there's no confusion here. It's not like I took these purines and I just hacked off a pyrimidine, okay? But that would be possible. So pyrimidines are cut from purines. One of the ways that I remember it too, because sometimes I can't remember which, if you don't remember that pyrimidines come first, that pyrimidines are cut from purines, okay, that sentence isn't very helpful to you. So one of the things that I think about is that the longer my word is, like for example, pyrimidine is a really long word. The smaller my molecule is. The shorter my word is, the bigger my molecule is. Okay, so big word, short one, 1 ring, small word, big one, 2 rings. Okay, so my pyrimidines are cytosine, thymine, uracil. So that means my purines are adenine and guanine. And so if you look at your base pairing rules, A to T, C to G, you'll notice that I have to have a pyrimidine with a purine. And that's going to be based on those intermolecular forces. I think we talked about this a little bit on the last video, right? C and G bond together because there are 3 places for them to make hydrogen bonds, and A and T only have 2 places. So it can't go, and A cannot bond with a C, even though adenine is a purine and cytosine is a pyrimidine. They cannot bond together because adenine doesn't have enough spaces to bond with cytosine. Okay, so like I said, there's hopefully probably only 5, maybe 10 minutes of stuff that is not reviewed from your freshman year. Okay, we'll be working with these more in class next time.